Welcome! Today we're going to talk about the amazing world of cells. You're going to learn about how we discovered cells, why cells are important, the difference between types of cells, and all the amazing things that make cells unique. So sit back, relax, and enjoy. Welcome back! So today we're going to talk about the cell theory, the history of cells, and why cells are so important. Not only that, but the two different types of cells. So let's get started. So first, we're going to talk about the discovery of cells, because cells and their discovery revolutionized science. We didn't really understand that there was a whole microscopic world out there until we discovered cells. And there were lots of people that made a very important mark on the discovery of cells in the microscopic world. And the first person was Zacharias Janssen. And if you'll remember, he was an eyeglass maker. And he was one of the first people to understand that when you put two lenses together into a compound lens, that you can magnify things very large. So he was one of the first people that made the compound microscope. And without the compound microscope, we never would have understood all of the realm of, the minuscule and the microscopic. The next great person was Robert Hooke. And Robert Hooke was a physicist, and he had many great discoveries, but one of his most interesting was his love of microscopy. And he put tons of things underneath of the microscope, one of which was cork. And when he looked at cork under the microscope, he saw all these tiny little structures that made him think about a monk's cell or a monk's room in a monastery. And so he termed these things cells. So in his first public work called Micrographia, he named what we now know as the cell. So he coined the term cell. And the way I like to remember that is Captain Hook put Peter Pan in a cell, because you'll need to know that for your quiz. The next person was Robert Brown, and Robert Brown was a botanist, and he looked at lots of different plant specimens under the microscope. And what he noticed, whether he was looking at grasses or flowers or shrubs, is that all cells had a very similar structure towards the center that was kind of darkish in its appearance, and he named that particular structure the nucleus. So he was the first person that named an organelle for the cell. And the last person who helped kind of understand the microscopic world was Anton von Leeuwenhoek. And Leeuwenhoek took it a step further, and he looked at pond water under the microscope. And what he saw in pond water absolutely amazed him. He saw all of these different creatures, creatures that had different kinds of appendages, and creatures that had different tails, and creatures that moved around differently, whether they had kind of short hair-like structures on their surface, or they had, tail-like structures. But all of them were alive because they were all moving. And so he termed these creatures animalcules, because he thought they were very animal-like in their structure, but they were microscopic. So he was the first person to look at living cells under the microscope. So that kind of opened up our world of discovery in terms of cells. The next great and important aspect in the history of cells was the development of the cell theory. And there are three people that were integral into understanding the cell theory. And the first person was Schleiden. And Schleiden was a botanist, and he looked at all different kinds of plants. He's German. And whether he looked at, again, grasses or dandelions or flowers, he saw that all plants were made of cells. He was good friends with this guy named Theodor Schwann, and they often had dinner together. And during one of their dinners, Schleiden was telling Schwann about the fact that he saw these structures in plant specimens that looked like cells, and Schwann remembered that he too had seen something very similar in the tissues of animals. So the two rushed back to the lab to confirm that also all animals are made of cells. So between the two of them, they discovered that all living things are made of cells. And in fact, Schwann published this discovery that all things are made of cells, all living things are made of cells, in his first book. And then came Virchow, and Virchow was studying leukemia. And leukemia, we know, is a form of cancer. And cancer is basically when cells have lost the ability to regulate cell division. So they divide and they divide and they divide and they multiply and they multiply and they form what we know as tumors. And so what he saw by looking at specimens of cancer cells, of these leukemia cells, was that in order to form a new cell, a cell had to divide. So he was the first person to understand that cells come from pre-existing cells, which was an important idea. In the history of the cell theory. So the way we can remember what each of these guys did is that Schleiden was looking at plants and came up with all plants are made of cells. So we like to say Schleiden was weeding for weeding plants. And Schwann came up with that all animals are made of cells. So we can think of that as Schwann's swans, so we liken that to the animals and the discovery of cellular structure in animals. And then lastly, with Virchow, well, there's no great way to remember that, so you're just going to have to know that Virchow was the one that came up with cells come from pre-existing cells. So let's go through each of the tenets of the cell theory, and there are 5 individually, and talk about what each of them really means. So first we have all living things are made up of one or more cells, and that's kind of an important distinguishing characteristic because some living things are just one cell, because we have this whole world of what we call unicellular organisms, the paramecium and the amoebas of the world. And then when we look at larger and more complex organisms, we also see that they are made up of cells as their basic building blocks. So even from the simple to the complex, all living things are made up of cells. The second tenet is that all cells arise from pre-existing cells through that cell division. And again, the only way that we realized that this was happening was by looking at leukemia. So when we looked at cells and we saw that one cell was creating 2 cells and 2 cells were creating 4 cells, we understood that in order to get new cells, we had to have an existing cell. So Virchow was really instrumental in understanding this whole all cells arise from pre-existing cells through the idea of cell division and understanding how cell division happens. Next up, number 3, we have that cells are the fundamental units of structure and function in living organisms. And this is really an important idea because for an organism to be really complex, its cells have to do all of these different functions. And what you're seeing right now is nerve cells. And nerve cells communicate with one another by sending electrochemical signals. And so that allows them to be doing their function and the function of organisms. You know, how we move and how we do our daily activities really depends on our nerve cells. So they are the basis of the functioning in living things. And then we can look at another example. If we think about the cilia that are found on some of our cells, those cilia do also amazing jobs for us. We have all of these cilia on our respiratory cells, and those cilia move mucus, and that mucus is really important because it traps things like bacteria and viruses in it. And then that mucus is moved from our mouth and our nasal systems into our stomach, where all of those bad things are broken down so that they can no longer harm us. So the idea that cells are the fundamental units of our structure and our function is a super important aspect of the cell theory. Next up, we have the idea that metabolism happens in our cells. And we know that metabolism involves enzymes. We have a big globular enzyme protein down here. And metabolism happens in our cells in the mitochondria. So the mitochondria is the site of metabolism, of us producing all of our chemical energy. And so the idea that metabolism happens in our cells might be new to you. We know that it happened in our body, but where exactly hadn't been something that we talked about yet. So the mitochondria and enzymes are really important to metabolism, and both of those things are residents of cells. Lastly, we have cells contain hereditary information, DNA, which is passed from parent to offspring during cell division. So understanding the fact that cells contain all all of the information on how someone is unique from someone else is a really important concept. And the fact that those particular genes can be passed on when we move from parent to offspring is a really, really important idea to the cell theory. So passing hereditary information is also a function of cells. So let's look at them one more time so that you can write them down and have them in one space. So first off, we have all living things are made up of one or more cells. From the simple to the complex, everything has at least one cell. Second, we have all cells arise from pre-existing cells. So in order to have one cell, you had to have a cell that it came from, and that's how living organisms reproduce and grow and repair themselves. Next, we have the cell is the fundamental unit of structure and function in all living organisms. From nerve cells to respiratory cells, all of those cells are helping us to do all of those complex jobs in our body. So they are not only building us in terms of our structure, they're also allowing us to do very important jobs in our bodies. Number 4, metabolism occurs within cells. So all of those chemical reactions that are producing energy and using energy are happening inside of different cells in our body. And lastly, cells contain hereditary information coded in DNA that's passed from cell to cell during cell division. So when we create a new cell, we have to make a copy of that information for that new cell, and that happens during that process of cell division. So that is the modern cell theory. So here we have a bunch of instances or different things that happened in the timeline of cells and their discovery and their history. So pause the movie for a second and see if you can put these in the right chronological order. So here's what you should have come up with. First, we had the invention of the light microscope. Without it, we wouldn't have been able to see any cells. Second, we had the discovery of cells, thanks to the help of the light microscope. And then we had the discovery of DNA. And in fact, we knew about DNA and we knew what DNA did way before we actually saw DNA. And then lastly, there was the invention of the electron microscope, which finally allowed us to see the elusive molecule that coded all of our genetic information, that was DNA. Have you ever wondered why cells aren't very big and why our body isn't just made up of a few very large cells? Well, part of that is because with many trillions of cells, we're able to lose some to sickness and to injury without having to worry about our overall health. But secondly, and more importantly, cells need to be small so that things that cells need to function in terms of metabolism and nutrients and then waste removal can go in and out of cells fast enough to sustain life. The larger cells get, the more volume they have in terms of relation to their surface area. So notice here, it's no matter where I would come into a cell across the cell membrane, it wouldn't take very long for me to get to the center of the cell. But as a cell gets larger, its volume grows, so I have lots more volume here than I do surface area, which is the outermost portion, or what my cell membrane is. And then when I get this large, I have way more volume than I do surface area. And in a cell this large, it just would be simply impossible to get nutrients in and waste out fast enough to sustain life. So the ratio of surface area to volume inside the cell is super important in terms of cells' efficiency and basically, how well cells work. So cells need a high surface to volume ratio for them to be efficient and for them to sustain life. If we didn't have that, we simply wouldn't be able to do all the metabolic functions necessary for our functioning. Let's move on to start talking about our different types of cells. So, if we think about evolutionary history, we started off with our very first cell, which we don't know much about. We know that it probably had a cell membrane and maybe some small proteins or pieces of RNA inside. But from that very first cell, we evolved our first basic type of cell, which is the prokaryotic cell. Prokaryotic cells were pretty simple, pretty small, not very complex. but they helped us to evolve all the other types of cells on our planet. So for that, they were very important. And we know prokaryotic cells as bacteria. So bacteria eventually allowed us to evolve eukaryotic cells, which are our more complex types of cells, and our second basic type of cells. Eukaryotic cells come in different types and shapes, but our two basic types of eukaryotic cells are plant cells and animal cells. So let's start first with talking about our bacterial cells, or prokaryotes. So prokaryotes have a number of different characteristics. First, we know that they are pretty simple. And what I mean by simple is that they don't have any membrane-bound organelles. And by membrane-bound organelles, I mean any organelles that are surrounded by a membrane. And some of our most important organelles are surrounded by membranes, and one of which that they are missing is the nucleus. So they have no nucleus. They also don't have any Golgi apparatus or mitochondria or any other of those other types of organelles. So what do they have? So let's look at the parts of a bacterial cell. So if you'll notice, they do have DNA or genetic material, but it's not in a nucleus. It's nothing that would protect it. It's just hanging out in the cytoplasm. So they also have cytoplasm, and then all of these little green dots are ribosomes. So while they don't have membrane-bound organelles, they do have ribosomes, which are not covered by a membrane. They have many layers of protection, the first of which is the cell wall, and they have a cell membrane on the interior, and some of them even have an outer layer outside of the cell wall called a capsule, which is just a third layer of protection from bacteria, because bacteria live in some pretty harsh places. They have a flagella, which is used for movement. It helps them to propel themselves through their watery environments. And then they have what are called pili, not to be confused with cilia. And pili are short hair-like structures that come out of the capsule or the cell wall of bacteria. And pili allow bacteria to adhere or stick to different parts of their environment. You have a lot of bacteria in your intestines, and they can stick to the walls of your intestines thanks to these pili. So they're pretty simple. That's about all that they have. They evolved 3.5 billion years ago. That's the oldest fossil that we have of bacteria. And they're pretty small in terms of the other types of cells. They're much smaller. And they are only bacteria. There's nothing else that can call itself a prokaryote but bacteria. So that's about it for our prokaryotic cells. Let's move on to talking about how we went from prokaryotic cells to eukaryotic cells. So eukaryotic cells, again, are much more complex. And one of the ways that they are more complex is that they have a lot of organelles. So exactly how did they get these organelles, or these mini organs? Well, one theory is the theory of endosymbiosis, which was put forth by a woman named Lynn Margulis. And she hypothesized that what happened was a larger bacteria basically engulfed a smaller bacteria. And the two of those bacteria actually lived in harmony with one another, or what we call symbiosis. It's a relationship between two living organisms until they eventually became the same organism. So we can think of this as an into a relationship. So there are two different basic types of eukaryotic cells, are plant or animals. So depending on what this little bacteria did that was engulfed to get its energy depended on what cell it eventually evolved into. If this little bacteria was photosynthetic, it most likely became a plant cell millions of years later. Whereas if this little bacteria performed cellular respiration, it became an animal cell millions of years later. In other words, this little bacteria, if it was photosynthetic, later became the chloroplast of a plant cell. And this little bacteria, if it was, a proponent of cellular respiration, eventually became the mitochondria of an animal cell. Pretty interesting thought there. So let's talk about eukaryotic cells. So one of the hugest difference between prokaryotic cells and eukaryotic cells is that eukaryotic cells have membrane-bound organelles, or organelles that are surrounded by a membrane. So the nucleus has a membrane, the Golgi apparatus has a membrane, the mitochondria has a membrane, the ER has a membrane, the little lysosomes have membranes. All of those organelles have membranes so that they can separate the interior components of that organelle from the exterior part of a cell. So eukaryotes are more complex because of all of these different organelles, and they're also larger than our little prokaryotic cells. They evolved 1.5 billion years ago, and they have a nucleus because they have the membrane-bound organelles. So all in all, eukaryotic cells are bigger, larger, more complex, and have many more different functions and things going on inside of them than our prokaryotic cells. And organelles are really the key to the complexity. We can think of organelles like rooms in a house. So in your house, you might have a bedroom, you might have a bathroom, you might have a kitchen, you might have a living room, and in each of those rooms, you're doing something different, a different function. You might be sleeping in your bedroom, you might be showering in your bathroom, you might be cooking dinner in your kitchen. And the nice thing about rooms is that they compartmentalize your functions. In other words, if I was cooking something in the kitchen and you were trying to sleep, our activities probably wouldn't impact each other because we are separated into these small compartments. It's the same idea in a cell. Organelles allow compartmentalization. Especially of functioning. And so when we have the mitochondria over here doing all these chemical reactions that produce energy, it doesn't really impact the nucleus, which is doing something completely different. So the organelles and the fact that they are separated from one another by membranes allows the cell to do all these complex jobs without impacting one another. So it's really helped us to evolve much more complex organisms, which really helped diversity on our planet. So let's complete a little Venn diagram, which would probably be really good to put into your notes about the differences between prokaryotes and eukaryotes, and also their similarities. So prokaryotic cells we know are pretty small, whereas eukaryotic cells are much larger. Prokaryotic cells, no membrane-bound organelles, whereas we do have membrane-bound organelles in our eukaryotes. It helps them to be complex. So we'll add complex over here, and we'll add simple over here. We have prokaryotic cells evolving 3.5 billion years ago. We have eukaryotic cells evolving 1.5 billion years ago. Prokaryotic cells don't have a nucleus because, again, they don't have membrane-bound organelles. And eukaryotic cells do have a nucleus. So small versus large, membrane-bound organelles versus none, simple versus complex, much, much, much older, much more recent, no nucleus, nucleus. Prokaryotic cells are only bacteria, and eukaryotic cells are every other type of cell. So what do they have in common? Well, all cells have a cell membrane. All cells have DNA. All cells have ribosomes. And all cells have cytoplasm. All right. Hope you got all that down, and this concludes your vodcast on the history of cells and the basic types of cells. I hope you enjoyed.